talk about actomyosin pulsation. Well, first of all, what are they? For our purposes right now, it's enough to know that actin is a protein that forms filaments, and myosin is another protein that is able to bind and unbind from actin and advect on it using ATP energy. This gives rise to pulsatile systems that are ubiquitous during development, like the ones you see here in the C. elegans embryo, and more relevantly for us today in the fruit fly Drosophila, the amnioserosa tissue of which you see here with the myosin in shades of grey and the cell membranes in blue. And here is a cell from the AS, and you see that the myosin forms these traveling waves called foci that contain almost all of the pulsatility of the myosin signal present in the cell apex. These foci can be identified and tracked using our very own homemade algorithm. And this leads us to observe a wide variety of focus behavior, such as focus birth and death, merging, and surprisingly, the splitting of foci. Now, more importantly, what can we do with these tracks? Well, for one, we can make a systematic observation of focus behavior, and this will be important later. Our observation starts with the increase in the average apical myosin signal in the cell, which is followed by the formation of one or more foci at subcellular regions. These foci travel on the apical cell surface until at some point they end and the foci die. Secondly, we can also use our tracking to approximate the foci as point particles and determine their speeds, their directions of motions, as well as the mean square displacements of focus trajectories. In short, we find that the foci move much faster than the substrate in a well-defined path, so their motion is not random, but we see that it's not ballistic either. To investigate what focus motion is really like, we want to answer this question first. How does the focus interact with its myosin environment? And to do that, let's think of the simplest focus possible, this one. Since we know from tracking what its coordinates of birth are, we can put our scope there and look radially outwards in this green circle before and after its birth, polling at each point the radially average and the maximum myosin. And this is what that looks like on the amnioserosa, averaged over about 2000 real focus births. Taking the maximum, we notice the presence of these depletion regions local to the focus before and after focus presence. This, plus the directional nature of focus movement and the non-ballistic exponents of motion, tell us that the focus motion is self-avoiding. But we can go a step further. If we look at the myosin environment of the focus just outside the cell it's in, we see this high concentration of myosin just outside the cell membranes. Since we know that individual myosin molecules cannot penetrate the cell membrane, we can say that the presence of focus leads to transcellular myosin recruitment. Now, knowing what we know, we return to our observations and posit a phenomenological mechanism of focus behavior. The transcellular communication could be a mechanism by which foci are born even inside a cell. Then this focus moves because of its self-avoiding nature, leaving a refractory wave behind it. And then it dies when it's trapped by the cell membrane and its own refractory wave. Now I'm sure that you have lots of questions, but for that and for perspectives on this work, please visit my poster. Thank you.